I start with giving you some background of what we do and why we do it, because I think for the younger generation of scientists, this is very useful in how to develop ideas, how to penetrate your dream experiment, and how to be able to perform it at the end. And I would like to finish with my dream experiment at the end. But before that, I'd like to introduce some of the people who contributed essentially to the data generation and interpretation of the stories I'm about to show you, especially Gabor Molnar, who is a senior electrophysiologist in our group. He is leading the in vitro experiments, and Robert Averkin is the sort of second leading electrophysiologist on the in vivo front. And I'm especially thankful to Professor Paul Barzo, who is our neurosurgeon at the Institute. And we started working in 2004 on human cortex. Now, the central question I would like to address in the first third of my talk is how to tackle the function of individual cell types. Now, a really simplistic example, and I think a useful example, is the pancreas, from a brain scientist. Just to give you a perspective, only 5 cell types, 5-6 cell types at most, compared to 150-ish of so-called saturated cortical cell types. Easy. Now, there are 2 approaches to tackle a function of a cell type. One is basically an unbiased method. You have basically a factory, you have hundreds of workers doing experiments, analysis, everything, and you're bound to produce results. The other, and this is the school I'm coming from, is the targeted approach. Limited resources, very dedicated people, unthinkably difficult experiments, and then you try to achieve the unthinkable in decades of work. And I'm a dedicated person, and I don't mind putting decades into something I really want to achieve. The problem regarding cortical cell types that they are not stable. They are dynamic. As we work, as we think, as the system develops, the function of a particular entity can change. I'm not saying it will change, but it can change. And this is a fundamental problem you bear in mind when you plan an experiment. Let that be an anatomical experiment. We touched upon anatomical dynamics in organotypic slices, for example. The molecular fingerprint is very dynamic. And we seem to forget that when we look at, transcriptomic maps of anything. It's very dynamic. Not to mention electrophysiology, which is actually targeted at this very dynamics. So this is an example of 1 year's sample collection from human brains at our site. We usually cover these areas, temporal the most, and definitely not primary sensory and motor cortices. Our sample size is limited, but we already passed 600 patients in these almost 20 years of work. And now we are, gearing up for more and more, the higher throughput. What's the basic methodology we started off with? 20 years ago, this was the standard we followed in our lab. Multiple recordings, some pyramidal cells, interneurons, connection testing, light microscopic reconstructions, electron microscopy of the identified synapses. Now we added some molecular biology on top of this, bearing in mind that we wanted to analyze identified cells, identified anatomically, electrophysiologically, and also molecularly. But on the molecular side, we don't want to compete with the big guys in terms of all-out transcriptomics. We want to be specific and we want to pursue the genes, the exact genes. We have some ideas from physiology or anatomy to be attentive of. And to that end, we developed a method which we call single cell PCR, digital PCR, with the detection threshold of a single mRNA molecule per cell. That's the precision. Now, when actually we looked at hundreds of specimens of our human data set, it turned out that we sort of bumped into cell types we haven't really seen in rodents. These are just initial examples of cell types we haven't really looked into detail, but 10 years ago we felt that these are potentially human-specific cell types. And that was the time when a chance of luck basically struck, and I got invited to be the member of the advisory board of the Allen Institute in Seattle. And actually, I initially helped them to establish the human workflow. And this membership later turned to collaboration. So I had to leave the advisory board of the institute, but I'm still, I enjoy work way more than sitting on committees. So ever since we are working on the human cortex together, and also joined by other excellent scientists sitting here. So, so what's the sort of the unbiased approach we developed initially together with the Allen people? Is basically you dissolve pieces of the brain, you isolate nuclei, and you do all-out transcriptomics on individual nuclei. And then you try to sort of organize that with statistical methods, try to delineate the clusters. Now, at this point, I would like to sort of focus on one problem which seemed to infect a lot of studies. Now, if you are interested how many cell types there are in a particular region, you can apply many methods. And usually people apply a mixture of methods to identify those cell types. I think that's a serious mistake, because you have to apply the same methodology throughout in order to gauge the diversity, the relative diversity of what you're sampling. So we actually paid particular attention to this. The first project we completed together was basically the layer 1 of the human cerebral cortex. And at that level of resolution, it turned out that we humans have, significantly more cell types residing in layer 1 compared to the mouse, like 11 versus 4 each. This is by no means saturated approach, but this is, just to give you an idea how much more diverse we are compared to rodents.
This is based on genetics?
Yeah, this is pure transcriptomics and unbiased approach. Now, obviously, the unbiased approach is not, to our liking really. So we felt that we need something for functional analysis. Obviously, we learned how the big data-producing scientific sites would do this, based on, for example, this Tasic et al. Paper coming out from the Allen Institute, and they generated, hundreds of cream mouse lines, and approached them, with targeted physiology, generated a huge data set, and claim that they sort of saturated the number of cell types studied or to be identified in the cortex. Obviously, having 15 people in the lab at most, we cannot compete with that, and we don't want to compete with that. So what we came up with is an alternative, targeted but automatized approach to counteract throughput problems. And what we did, actually, we teamed up with a computer microscopist at the research center next door of the Hungarian Academy of Sciences. And actually, the group of Peter Horvath was extremely useful in coming up with an image stack-based solution of recognizing cells in DIC, Z stacks. Cell recognition of the artificial intelligence is better than Gabor Molnár's, mine, or one of my PhD students here. Now, then we actually developed a complete system for automated patch clamping, which can actually recognize cells in the way of reaching the target neuron, and it can actually pull back the pipette, go around it, and target the cell we wanted. So here's an example, how it works. So this is the automatic system. You have the slice under your scope. You generate the Z-stack. Your cells are being identified and recognized. This is the cell we want to patch. And the system actually positions the pipette automatically. Pressure on, approach, target the cell, release the pressure, form the seal, break in, and then you have whole cell. This is the real example how actually it works live. So there's the dimple on the human pyramidal neuron there. You see seal formation in this panel, break in, and then firing. Firing, which is identical to sort of manually patched cells. Anatomical features are also very useful. So this actually sort of tripled or quadrupled our output in terms of single cells. And this was very instrumental in uncovering unknown cell types in layer 1. The first unknown cell type which we focused on was the so-called Rosehip cell type. Actually, we named the cell based on the morphology of the axon, and not just the bush-like, really compact axon, but based on these individual Rosehip-like boutons, which are extremely densely intermingled in layer 1, and at least at the time we wrote this paper, we could not find Rosehip cells outside layer 1. And if you compare Rosehip axonal morphology, for example, the Neurogliaform axonal morphology or basket morphology, I think the pictures speak for themselves. Now, what is the function of these Rosehip cells? And here is where I think the brilliance of Gabor Molnár comes in or shows itself. Now, here's a Rosehip cell, layer 1, and here is a layer 3 pyramidal cell. Gabor patched onto the Rosehip cell, found these IPSPs on the pyramidal cell, and then started to image the pyramidal cell dendritic tree at about 50 different points in order to catch the points at which the inhibition was happening. And out of those 50, these are 3 representative points, and only at one of the points imaged, he could see an effect. These other 2 points, he could not. And if you look at the position, here is the axon of the, here is the dendrite of the pyramidal cell, image spot number 1, 2, and 3. And we see the effect on point number 1, which is about 5 micrometers from the actual synapse between the presynaptic axon and the postsynaptic dendrite. But we don't see effect 5 and plus 5 micrometer further apart. So this is a real local inhibition of backpropagating action potentials we gauged in response to inhibitory input from these cells. So what these Rosehip cells do, actually they provide extremely precise, spatially very compact inhibition to distal dendrites of layer 3 pyramidal cells. The reason I'm pressing this pyramidal cell connection is that it took us more than 3 years to find the first connected pair to a Rosehip cell, because we were looking for potential partners in layer 1. And in layer 1, there are no pyramidal cells. And you need pyramidal cells to get potential postsynaptic partners.
You're saying, just to make sure you understand, there is a back-of-again action potential of green cell,
yeah,
pyramidal cell. Then you measure the calcium of this location,
yes,
from this back-of-again
action potential.
Then you activate the Rosehip cell, see a reduction on the calcium of this location. This is the inhibition.
Exactly.
Of the back-of-again action.
Exactly. Exactly. And if you are more than 5 micrometers apart, there is no effect. It's a microdomain inhibition. But my favorite cell type is not the Rosehip cell type, I have to admit. My favorite cell type is the Neurogliaform cell, discovered by the great Ramon y Cajal. And the name is derived from the extremely dense axonal arborization, which actually resembled Cajal to astrocytes, the dense meshwork of astrocytes. We actually bumped into these cells, I don't remember when, type of temporal distance. But at that time, the problem was that although it was discovered that in the neocortex, you had 2 kinds of inhibition, fast inhibition through GABA A receptors and slow inhibition through GABA B receptors. And then approximately 15 years after this crucial discovery, we found the first cells which could actually reach GABA B receptors. And these cells turned out to be the neurogliaform cells, which could combine GABA A and B receptors, while the rest of the GABAergic cells reached only GABA A receptors on postsynaptic neurons. And so far, the neurogliaform cells are still the ones which can do this in response to a single presynaptic action potential. Now, how do neurogliaform cells do this? When GABA is released from a terminal, it actually covers an area of about 3 micrometers in radius with efficient concentrations of GABA. So in case you put 2 separate axonal branches closer than that distance, chances are that you can flood the area with efficient concentrations of GABA. Now let's have a look at how many separate axonal collaterals we have, let's say, at this corner here. 1, 2, 3, 4. And the distance between these 2 is less than 10 microns. So the density of a single neurogliaform axon is compatible with the idea that the axon is flooded by GABA and there is a volume transmission sufficient to cover everything. So the way a neurogliaform cell works is like a Chinese noodle soup. So this is the axon, the noodle is the axon, and the GABA soup is provided out of the axon. And no matter where you put your spoon into your soup, it will get wet, right? So this is the way it works. But this is a very interesting, idea, because everything having GABA A, B, or whatever GABA receptors will get that GABA, no matter whether it's a neuron, an astrocyte, or whatever component of the system. And actually, we managed to show that not only neurons, but glial cells also respond to individual neurogliaform cells. And perhaps more importantly, blood vessels also respond to neurogliaform cells, or perhaps even more importantly, presynaptic terminals also respond to neurogliaform cells, yet you do not have synapses on presynaptic terminals in the cortex. So you don't need synapses. You need a neurogliaform cell to flood the area with GABA, and you will get a response. So, and that response is, you can call it volume transmission, but you can also call it tonic or ambient GABAergic inhibition. It depends, which school you are coming from. So in a couple of years ago, we were really intrigued by your work, actually, showing really nice spatial distributions of axons and their workings in the cortex. So what we wanted to do is to have an idea about the population output of all the neurogliaform cells in the cortex. So what we did, we combined individual 100x neurolucidary reconstructions. This is the original 16 neurogliaform cells, which we used as a template. We generated so-called a model output axon for our purposes, which was in good agreement with the original reconstruction of 16 cells. And then we did a staining, which would correspond pretty much to the 3D positions of neurogliaform cell bodies, and placed these ideal individual neurogliaform axons, modeled axons, with centered around these positions, and came up with this complete output of all the neurogliaform cells in a region of the cortex.
When you say complete output, what do you mean?
Meaning these are the axons of all the neurogliaform cells residing in the area. Okay. Now the aim was to have an idea how dense the output is. Namely, in case you pick a random point, is there a neurogliaform cell nearby to be able to cover that area? Now, the question is, yes. There is no point where there isn't a single one providing GABAergic output to. In fact, you have either 1, 2, 3, and 4 at most simultaneously covering the same randomly chosen point. So up to 4 neurogliaform cells might reach the same point, but every single randomly chosen voxel is covered by at least one. So the blanket of GABA is complete, so no one escapes it.
All through 6 layers?
This is layer 1 and 2, what we covered here. 1 and 2. Now, what percentage of those neurogliaform cells will be active? And we actually gauged this with a simple, relatively simple in vivo experiment. What we did actually, we stimulated the hind limb while imaging with normal, low temporal resolution OGB imaging layer 1 and 2 with 2 photon methods, mapped the position of all neurons in layer 1 and upper layer 2, and then made a counter stain of the very same area with the neurogliaform marker, and then sort of post hoc identified the cells which turned out to be neurogliaform from the imaged ones. Now, it turns out that in response to this in vivo stimulation, about 40-ish% of all neurogliaform cells respond with detectable calcium signals. So about half the cells can be simultaneously active, meaning that if half the cells are active, then in vivo, the situation is very really, a complete coverage of the area with effective concentrations of GABA. And this opportunity actually be used to gauge how signals generated by neighboring neurogliaform cells would sum up. Because, when you're looking at the literature, we found that only ionotropic receptor summation was studied before. Metabotropic receptor summation somehow sort of left out of this approach. And so what happens here, just to wrap up this part of the story, the GABA A component sums sublinearly, slightly sublinearly, but the GABA B component linearly. And I can be quite sort of categorical in this because I know from the anatomical modeling part that there is no more than 2 cells active at any time at any cortical voxel, presynaptic cells. So this is a saturated activation of the system. So the saturated activation would lead to slightly sublinear GABA A and linear GABA B summation. So there is no amplification of metabotropic summation by whatever amplifying coupling between, the G proteins and the effector potassium channels here. But we were also curious. Any question?
You mentioned voxel. What
Voxel is like a randomly, what size?
Yeah, when you say
Our
1.2 per voxel, then how large?
The voxel is any randomly chosen point, and I'm talking about a radius of the effective GABA concentration coming out of so 3 micrometers. So that's my sort of gauge in the system. So, but how about the transcriptomic identity of neurogliaform cells compared to the rest of the brain? So what differentiates neurogliaform cells from the other cell types? But to that end, we did this 10 years ago and still working on some of the aspects. So we collected about 40 neurogliaform cells. We identified based on postsynaptic effects. So just to be sure that these are GABA B eliciting cells. Took out the cytoplasm, collected all 40 of them, and did complete transcriptomics on those 40, because at that time we couldn't do it on single cells. Nobody could. And then we compared this to the average transcriptomics of the surrounding brain tissue and ranked the genes based on different expression levels. Now, number 6 on that, difference ranking turned out to be insulin. That time I felt that, how come that insulin has anything to do with, interneurons or the cortex in general? And it turns out that, insulin has, a set of really important functions in the brain. And here's this classic paper showing that insulin concentration in the brain is 10 to 100 times, I repeat, 10 to 100 times compared to the blood. Yet, we do not know whether insulin is being produced by the brain or not. In fact, there's a huge debate going on, or was going on, whether insulin is, little or no insulin is produced in the brain, or there are cells in the brain which would be positive for insulin mRNA expression. But, insulin readily crosses the blood-brain barrier. But in case there is no mechanism to enrich insulin in the brain, then, it doesn't work like that. So you need something. Why is this important? Because if you look at people, at initial stages of Alzheimer's, it turns out that, there is a decreased cerebrospinal fluid, blood plasma insulin ratio in those individuals. And this observation actually triggered a renaming of Alzheimer's disease in the literature, type 3 diabetes. So this was the point when I decided, okay, we are onto something potentially interesting. So what we did, actually, we compared the insulin expression of individual pyramidal cells and neurogliaform cells with single mRNA resolution. And it turned out that in neurogliaform cells, we have about 30, pyramidal cells about 7. And this actually responds to glucose concentrations outside the cell. So the higher the glucose concentration, the higher the insulin expression level by neurogliaform cells. So this is the dependence of glucose concentration dependent insulin expression in neurogliaforms. Glia, pyramid, other interneurons do not respond. But what insulin does in the cortex? Controls you have a lot of spontaneous EPSPs. Insulin suppresses those. And then if you block insulin receptors, spontaneous EPSPs come back. So how it's done? Insulin actually takes internalized GABA-A receptors out from, internalized membranes to the plasma lemma. And so it increases the tonic GABA-A current. So that's why you actually can suppress overall excitability. So based on this, we planned an experiment with a classic, pharmacological approach in mind, that we have to show that insulin is being released from neurogliaform cells. So we actually puffed glucose onto a neurogliaform cell, and we monitored in a neighboring interneuron the spontaneously occurring EPSP frequency. And we could show that relative to control, glucose puffed onto another cell, the neurogliaform cell, could suppress EPSPs arriving to this cell here, and this suppression was sensitive to insulin receptors. And this is the classic way of showing a neurotransmitter in action, because you show that it's being released, and you have an antagonist, a selective antagonist with receptor, and you can block it. Yeah. Yeah. Tens of minutes. So it's relatively fast. Tens of minutes. So basically, this is the schematic representation of this experiment. We puff glucose, insulin is being released, you can block it with an insulin receptor antagonist. And this happens to be, the response to not just glucose, but to other insulin release triggers, like a classic way of suppressing initial phases of diabetes is the application of sulfonylureas. So in case you apply sulfonylureas instead of glucose to neurogliaform cells, same thing happens. But with the help of sulfonylureas, we could actually show directly that if you apply sulfonylureas into acute brain slices, so taken out of the brain, we could increase the insulin content of acute slices, showing that regardless of whether the blood-brain barrier is intact or not, local insulin synthesis can be detected directly with the radioimmunoassay. So this is a direct proof that we can increase insulin content using, the most widely described anti-diabetic, sulfonylurea, which, by the way, crosses the blood-brain barrier too. So now at this point, we also felt that we should really push this into the human direction. And the next step, what we tried, was the so-called incretin effect. You are not cretins not knowing what the incretin effect is. So in case you consume a meal, what happens is that in response to that, you produce GLP-1, and that GLP-1 boosts insulin release from your pancreas. Okay? This is called the incretin effect. So GLP-1 binding to pancreatic beta cells boosts insulin release. Now let's see whether, neurogliaform cells can do this, because, this incretin effect apparently is hampered in half of the European population being overweight. So people with, on the way to type 2 diabetes have a suppressed incretin effect. So in case we can manipulate this, it would be hugely beneficial for society. And we figured that let's just probe whether insulin and GLP-1 is expressed in rodent and later on human neurogliaform cells, with the hope that GLP-1 receptors are present in neocortical interneurons in a sporadic manner. So let me just, broaden your horizons a little bit. So why GLP-1 and this problem is important to a neuroscientist as a general perspective? So cortical output is really important in the nucleus accumbens, where all sort of abusable substances seem to have their effect, right? So no matter whether you take heroin, nicotine, alcohol, amphetamine, it will influence dopamine release in this area. Now that dopamine release, in case that dopamine release is coupled with glutamatergic excitation arriving from the cortex, then you are done. Okay, so that's why, yeah. So, so that's why we felt that, okay, now let's have a handle on this let's have a handle on this cortical glutamatergic output in a way of potentially manipulating neurogliaform cells. The first experiment we did, we showed that GLP-1 receptors are present in rodent neurogliaform cells, and more than that, they are present according to the external glucose concentration. So if you lower the glucose concentration, copy numbers drop to about 2. So, and the GLP-1 receptors are co-expressed in neurogliaform cells with insulin. Now, this is a very interesting sort of cell biological experiment because, notice that from about 8, we drop the GLP-1 receptor level to about 1 or 2 in neurogliaform cells. And electrophysiologically, we can uncover whether 1 or 2 copies of a particular mRNA would produce a functional response. And indeed, when we had more than 2 or 3, we could actually block GLP-1 receptor effects. But in case we dropped below 2 mRNAs per cell, no physiological effect. So this seems to be sort of the lower boundary of production to produce an electrophysiologically meaningful effect of a particular gene. And to my knowledge, this was the first experiment which would actually show this number. So you don't need a lot of copies to be effective at the protein level. So to wrap up, the last 10 minutes, what I try to convince you about is that neurogliaform neurons are, very similar to pancreatic beta cells, but they reside in the brain. Indeed, developmental genes of pancreatic beta cells can be found in neurogliaform cells too, and all the key genes can be found there. Now, how about human neurogliaform cells? Now, to this end, we again needed to develop a new sort of methodological approach, because first we need to identify what type of cell we are dealing with in an acute human setting. And then we have to count the number of mRNAs of a particular gene, in this case, insulin and GLP-1 receptors, in the identified cells. Now, to that end, we actually looked back to our paper showing neurogliaform cells, for example, but also human-specific cells, and tested if, using like 1/5 of a sample of an acute surgically removed specimen, circumcised by a laser beam from a frozen specimen of human cortex, a single cell is taken out. We use 1/5 of that cell to identify the cell type itself, and then the rest we use for digital PCR. And we are in position to show that human neurogliaform cells are also insulin positive. Human neurogliaform cells also produce GLP-1 receptors, and not just neurogliaform cells, but other layer 1 interneurons found only in humans, by the way, also produce this. And other interneuron types lack insulin, but express GLP-1 receptors. Now, this effort actually shows you that evolution in novel cell types in us can contribute to essential functions. So there is a multiplication of functions, which I consider very important. Now, what would neurogliaform cells do? At least my current view about neurogliaform cells is the following. You can actually really broadly set the excitability level of the neighboring microcircuit by flooding or not flooding the circuit with GABA, including glia, blood vessels, and so on. Now, this is achieved together with insulin secretion, and insulin secretion can be controlled by GLP-1 receptor agonists, which are the most widely described anti-obesity drugs today. And with that handle, what we are suggesting to clinicians these days is that we should really see those clinical trials which will uncover this potential effect. Because you can deliver insulin directly to the brain by puffing it into your nose. So there's a direct pathway of peptide transmission from the nasal cavity to the brain. However, you can locally boost insulin production by adding GLP-1 receptor agonists peripherally. Now, up to this point, I wanted to cover a couple of points. Taking this sort of natural setting to sort of mimic neurogliaform action, having my wife dangling in this spray of not GABA but water in this case. So I showed you that artificial intelligence-based approaches can boost human recording throughput. Out of layer 1 interneurons, there are some evolutionary conserved and novel cell types. And neurogliaform cells express insulin, GLP-1 receptors, and could serve as therapeutic targets for suppressing obesity in the long run. Now, I still have a couple of minutes left from the first part of my talk, so let me use that to introduce the last bits of my presentation. So let me return to this, really old approach of patching to one neuron and recording the effect in another neuron in the human. This slice happens to be from a 75-year-old male patient, by the way. What usually happens if you do these sorts of experiments is that a single presynaptic spike leads to a single postsynaptic response. Not in the human, at least not all the time. What you have is a single presynaptic spike and a bunch of responses temporally linked to that particular presynaptic spike. We know that these events are linked because the timing of inhibitory or excitatory polarity responses appears to be fixed relative to the presynaptic spike. Okay. Now, what about the temporal structure of this range of events triggered by a single episode? Is it like an avalanche of cortical responses, uncontrolled avalanche, or some sort of a sequence of motifs like this, Hungarian folktale cartoon type of motif generation? Obviously, being a Hungarian, this gave you the answer to this question. So you have a presynaptic spike, generates clouds of events, like an inhibitory cloud, an excitatory cloud, and then a sparser inhibitory cloud in pyramidal cell number 2, and a simultaneously recorded pyramid 3 receives 1, 2, 3 clouds of inhibitory events. And when you look at the timing of, let's say, an inhibitory event here and an excitatory event here, they shift together relative to the spike just a little bit. So there's a temporal jitter, but when there is jitter, they move together. So they move relative to the spike, but they don't move relative to one another. So the motive is fixed and it's triggered by a single spike, which means that we run into a classically described phenomenon called functional assemblies by the great Tamas. So these in the human cortex can be triggered by a single spike in a single cell, apparently. And this observation was so striking that we spent 15 years ever since to uncover, why is this so? First of all, let me show you a couple of examples. What sort of cells can trigger this and how many synapses are required for this? Here's, for example, an inverted pyramidal cell, apical dendrite, going towards layer 6 instead of the PM. These cells can also trigger. Now, pyramidal neurons. So just that I wanted to show you. So how many synapses you require? Here is a synapse, a single synapse linking pyramidal cell, axon coming out of the axon, arriving to the postsynaptic dendrite. And this is the effect. So 77% subthreshold response, 23% postsynaptic spike in response to a single synapse. Very effective, I think. So what makes pyramid to interneuron synapses so efficient in the human? Now, to that end, we applied a really tedious method of quantal analysis and systematically compared the anatomy and synaptic physiology of identified layer 2-3 pyramid to interneuron pairs in the human and in the rat. So here's an example of a presynaptic pyramid giving rise to this green axon, having 4 synapses on the blue interneuron dendritic tree. Same here, presynaptic soma axon, 2 synapses on the interneuron tree. Now, at this point, I would like to point out that the size of these 2 cells is not the same as these 2 cells, okay? Yet the latency appears to be similar. This will be important in the last part of my talk. But the only thing different in terms of electrophysiology is the charge carried through these synapses, the charge. The decay time, rise time, latency, not different. The charge is different. And when you do multiple probability fluctuation analysis, as I said, a heroic method for quantal analysis, if you consider that you have to change the extracellular calcium concentration 3 times in a human setting and having quality, high quality voltage clamp analysis, that's quite something, I believe, as an electrophysiologist. So anyway, what differs between a human and the rodent is the number of functional release sites in these synapses. So for those of you who are not familiar with quantal analysis, the number of functional release sites means how many vesicles are being released through a connection. So what we did, we serial sectioned excitatory synapses arriving to human and rodent inhibitory dendrites and counted how many dogged vesicles we could see in the human and in the rodent. And it turns out that we have about 5 times more dogged vesicles in the human compared to the rodent. Now let's just jump back a little bit. We have about 5 times different in the number of functional release sites gauged by electrophysiology between the human and the rodent. Now, then we went on and applied a method called electron microscopic tomography to increase our resolution and to measure directly the density of doped vesicles. So what you do, instead of cutting 20 nanometer thin sections, you cut a couple of 100 nanometer thick sections, and you can tomogram basically the insides of that relatively thick EM section. And this is actually just the tomogram of that thick EM section. And this is a synapse in that very section. The first sort of view is just, native. And then we marked the doped vesicles, the omega profiles in this synapse, so we could directly measure the density. And it seems that it's not only the number of doped vesicles, but also the density of doped vesicles is higher in the human compared to the rat. So we don't need a lot of spaces to put higher numbers of vesicles, doped vesicles, into excitatory synapses. So this is the profane presentation of what happens in a human excitatory output synapse. You are scalable and strong, or can be strong, in case you are human. But you are weak, and you either have a chance or don't have a chance, in case you are unhuman or not human. Very simplistic approach. So anyway, so we do have a choice, and our synapses can be stronger, scalable. Yep. So this is the first, so this is the trigger output of this Hebbian network. You have basically sort of strong synapses targeted towards interneurons and weaker synapses targeted towards pyramidal cells. Okay. Now, how about the generation of these downstream excitatory events? Over here. When you actually look at the sensitivity of these polysynaptic events following single human action potentials, the striking observation is that disynaptic IPSPs are sensitive to GABA-A blockers like gabazine, okay? But not only IPSPs are sensitive, but EPSPs are sensitive to excitatory polysynaptic potentials like these are sensitive to gabazine. And those are trisynaptic latency, right? Not disynaptic, but trisynaptic latency. The key to answer the puzzle is the so-called exoaxonic cell. What happens in an exoaxonic cell in response to a pyramidal input is either a big-ish EPSP or a postsynaptic spike. So like interneuron, strong synapse, postsynaptic spike. But there's more than one spike here. It's not just one, but two postsynaptic spikes. So let me clarify a couple of things about exoaxonic cells. They exclusively target the initial segment. Nothing else but the initial segment. That's one of the major points. Second, they don't target interneurons, just pyramidal cells. A very unique feature, okay? And in case you quantify the molecules which are responsible for setting the chloride reversal potential in a pyramidal cell, it turns out that KCC2, which pumps chloride out of pyramidal cells, is enriched perisomatically, but is absent at the axon. So there is nothing pumping out chloride, meaning that in case you had an input from a basket cell that's hyperpolarizing, in case you had an input from an exoaxonic cell, that can be depolarizing at the same membrane potential. We actually directly compare the efficacy of excitatory events arriving to perisomatic parts or to the axon of pyramidal cells, just to see how efficient these can be. Before that, we measured with our somatically placed electrode the threshold of action potentials in response to long-lasting depolarizing pulses, around 44 millivolts in both cases. When we sort of set these basket cell inputs to depolarizing by using high chloride solution inside, the threshold in response to these inputs was similar to the current injection triggered threshold, around 40-something millivolts. In the axon, the threshold is not around 40 millivolts. It's about 75-ish, minus 75 millivolts. It's way more hyperpolarized, okay? And we know that these inputs are on the axon because X-axonic cells place the inputs only at the initial segment and only at pyramidal cells. This will be important later too. So anyway, what about the output of X-axonic or X-axonic cells? You have pyramidal neurons with, postsynaptic chloride conditions, allowing hyperpolarization or depolarization. You have a subset of pyramidal cells which occasionally respond with spikes, and you have another subset of pyramidal cells which actually respond with polysynaptic excitation. It's very important that the ratios of pyramidal cells which would respond with an excitatory effect is very small. Most of the pyramidal cells receive an inhibitory input or shunting input from X-axonic cells, and only about 4% would receive a triggering spike. Okay? Now, let's revisit this single presynaptic spike, primary event, and two postsynaptic X-axonic spike in a network. So the primary event is here. Secondary event, a spike in the X-axonic cell, and a spike somewhere else in the network in an intended. Tertiary event is a spike in a pyramidal cell triggered by this X-axonic cell here. We know that there is a spike here because the X-axonic cell receives a trisynaptic input from this pyramidal cell, as shown here, occasionally a further downstream spike riding on top of that. So this is the mechanism of explaining how a single pyramidal cell would trigger trisynaptic EPSPs sensitive to GABA-A blockers, because this is the point you can actually block it. My late PhD supervisor, Eberhard Bull, showed the very same phenomenon in a paper almost 100 years ago, 30 years ago, not 100, 30 years ago. So if you look at this recording from an X-axonic cell, cat visual cortex, completely different experiment, right? What you see here is spike doublets in an X-axonic cell. And if there is no spike doublet, there is this, what he called excitatory post-potentials right after the spike, which are actually these EPSPs coming back from further downstream pyramidal cells. So, and here is the effect of a human X-axonic cell. Single spike, a network oscillation, and some downstream spikes arriving back to a human X-axonic cell. So a single cell can initiate a 200 Hertz rhythm of 2 full cycles in the neighborhood. Okay? So what happens here is basically a cycle of pyramidal cells, basket cells, pyramidal cells, basket cells, stop. BC, 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 like this, but at 200 hertz frequency, right? So, and that 200 hertz frequency happens to be extremely important in a memory formation called sharp waves, okay? And the sharp wave frequency is exactly the same as this single cell triggered so-called sharp wave at the human cortex, in the human cortex. And this was the point where we decided to do in vivo recordings in search for these local, focal, mini sharp waves in the neocortex. Because when there are sharp waves in the hippocampus, there is memory transfer to the neocortex during slow wave sleep. However, sharp wave ripples in the neocortex were not recorded. They were elusive. Now, let's think about it. How should this memory transfer work? And, you know, again, a profane everyday example, possibly. So you have a transmitter, like a radio transmitter, and you have a receiver. The key is that the transmitter and the receiver uses the same matching frequency, and you modulate the amplitude to transfer something, okay? Now let's search for this matching frequency in the neocortex. Maybe we find these 200 hertz ripples. And to that end, we developed the method of juxtacellular recording in freely behaving, completely undrugged, I repeat, completely undrugged animals, and found those 200 hertz ripples in the neocortex, and found them embedded into neocortical spindles during slow wave sleep. So what happens is that at each trough of a neocortical spindle, you have a sharp wave embedded, okay? And those sharp waves actually are centered at the spindle trough. And they have nothing to do in timing with the hippocampal sharp waves. So it's not a radiation of hippocampal sharp waves. These are locally generated. And here's an example how completely drug-free animal produces something at a cellular resolution which might be relevant to explain these neocortical sharp waves. Let me just play this again. Yes. So you see spindles here, and at the troughs of the spindles, you see these spike packets. Now the animal awakes, okay? Spindles disappear, and those spike packets also disappear, okay? So this is the analysis of this recording. You have this cell recorded juxtacellularly, and centered at the trough of this spindles, basically, you have these 3 bands of spike activity, and the 3 bands are separated by plus minus 5 milliseconds exactly at 200 hertz frequency. So there is an interneuron which follows these spindles at the trough with ripple frequency. Interspike interval peaks at 5 milliseconds. So this is the natural frequency of this cell. And it does not change over the course of a spindle. Now, if you look at the activity of this individual cell relative to the population, this is basically an almost in-phase activity, okay? Now let's have a look at pyramidal cells in the same setting. Layer 2 cells are not related to this. Layer 3 cells show something on the bottom of the spindle troughs, but their timing is shifted relative to the population average. This is a sort of the simplified explanation for this. You have the spindle trough with a 200 hertz ripple on top. Interneurons, at least some of them, are in phase with that ripple. Pyramidal neurons are anti-phase with that. That means that you have a reciprocal synaptical communication between the two at 200 hertz. It turns out that this is a monosynaptic loop back and forth. So it's a PCBC, PCBC, PCBC 200 hertz, what you see here. So we would like to propose that these are evolutionary conserved motifs of the rodent circuit and the human circuit, monosynaptically connected pyramidal cell, basket cell, pyramidal cell, X-axonic cell, pyramidal cell loops producing these neocortical ripple-like events, which might be helpful in memory transfer. So there are some salient features you can notice in case you are off the coast of South Africa. And I actually chosen this picture of mine just to entertain Henry. So, and those features are the strength of human pyramidal output synapses can be huge on interneurons and weak on pyramidal cells. These synapses can drive postsynaptic cells to fire. So there is spike-to-spike coupling. Ripple-like oscillations occur at the trough of sleep spindles. And these ripple frequency oscillations are sort of composed of alternating pyramid and interneuron firing and can be observed in the rodent and also in the human. And I thank you for your attention.